All right, last part of today's lecture. And we just have to remember or remind ourselves that synapses that we are learning about, right, can be between a neuron and some other cell except a neuron, right? In the first case, that's most commonly kind of perceived that a neuron 1 will have the cell body with all the dendrites, and then it will send, this axon with axon hillock down, or to the kind of postsynaptic neuron, right, where the synaptic endings or terminal will end up in the synapses that we learned about, right? And this is how information will be conveyed, including our reflex that we just learned, right? From the sensory neuron to the lower motor neuron or towards the interneuron, okay? So from afferent to efferent, right? In the second case, right, and these are the two diseases that we're going to mention today, is where a neuron, right, that lower motor neuron, the efferent neuron that is having a cell body in central nervous system, but the axon is coming out into the peripheral kind of part of the nervous system, right, is going and basically sending that axon, right, out, and touching the skeletal muscle fibers, right, the muscles, the extensor, the quadriceps, for example, right, or flexor of the hamster. All right, so, and these synapses are called like neuromuscular junction because it's between neuron and muscle. So this is going to be called neuromuscular junction, okay? The third type, and we're going to talk about them when we come to autonomic system, are the neurons that will have these axons going towards the glands to secrete certain hormones or saliva, and so on and so on. And they are called neuroglandular synapses, all right? Okay. You have to also remember, and that's important for pharmacology, right, that some neurons will also contact directly the blood vessels and secrete some hormones into these blood vessels and so on. And we will touch upon that too. It is just kind of to keep that in mind too, that beside these three, there is also direct contact with some blood vessels and some neurons, okay? So let's get to the meat of it, to the neuromuscular junction, okay? So here what we can see in this picture, right, the purple ones are the muscle fibers, okay, coming down. In the dark stain, we have this axon coming down, right, from that, from the spinal cord. The cell body is in the spinal cord, remember, and this axon is leaving the spinal cord and going and traveling towards the muscles. It can be that extensor, flexor, whichever muscle we want to focus in the skeletal system, right? And these motor axons will then end up, on these fibers. You see the fiber as motor end plates. So that's kind of a $10 name for the neuromuscular junction, where it is happening at the motor end plate, okay? And that's, again, a chemical synapse, right? There is a neurotransmitter that is a chemical between this efferent neuron and the muscle fiber that is going to be receiving those neurotransmitters, okay? So just this term that we introduced, the motor end plate, right, is kind of a $10 word for neuromuscular junction, okay? So we don't get confused if the question is talking about motor end plates. We are actually focusing on the axon, presynaptic terminal, and the muscle as a postsynaptic membrane of a neuromuscular junction. And here is what we are going to now do. We are going to introduce the first neurotransmitter of our journey in the neuroscience, okay? And this one is called acetylcholine. Remember all those chemicals in synaptic vesicles, one of them is called acetylcholine, okay? And it was first discovered in this neuromuscular junction, and it is very well studied because of this, of the kind of approachability, so to speak, at the neuromuscular junction at the periphery, okay? So basically, the neurotransmitter is a chemical message, right, in specific, with specific characteristics, that is released from this presynaptic motor axon, and is looking for its receptors on the muscle fibers, okay? Once it was discovered in the neuromuscular junction, further studies also revealed that acetylcholine is also present in the central nervous system, okay? And we're going to talk about them when the time comes. Today, we are focusing only on the neuromuscular junction, okay? Again, in this upper right corner, you can see muscle fibers, these axon terminals with these motor end plates, right? And now if we zoom in, we are already familiar with this. We know it, okay? So we're just going to give some names to it, okay? Again, we have this presynaptic terminal. You see this axon coming down, right? It has these calcium channels, right, in this presynaptic terminal. It has these synaptic vesicles. And in this case, these red dots, they are neurotransmitters, but now we know the name, right? It's acetylcholine, because we are talking about neuromuscular junction. In this case, you can see we can even see this myelin sheet, the Schwann cells, because remember the cell body of this motor neuron is inside the central nervous system, but the axon is at periphery. So who will provide the myelin for the lower motor neuron or efferent neuron will be Schwann cell. Okay, so let's not confuse them. Even though cell body is central nervous system, the myelin is provided at periphery. So it is a Schwann cell that is giving that myelin insulation for this presynaptic terminal that is trying to reach the muscle. Okay, so now let's walk through it, and you already know it. Okay, you will see how much you know. So basically, the action potential is coming down. Remember, through this axon, is changing the voltage, right, of the membrane. The calcium channels will open, which means the calcium that is outside, and based on its electrochemical gradient, it will rush in. This calcium will go in and move the synaptic vesicles with acetylcholine, right, towards the membrane, right? And this acetylcholine will be released into the synaptic cleft, and acetylcholine will now search for its receptors. You see on this muscle as a postsynaptic membrane, right? And this kind of binding of acetylcholine to the postsynaptic receptor, the acetylcholine receptor site, will allow the sodium to come in and depolarize the membrane of a muscle, okay? So not to overwork the muscle, we have to get rid of acetylcholine, and here is that enzyme. You know, remember that one that is cutting the acetylcholine in two pieces, acetyl and choline. That's why it's called acetylcholine. And this enzyme is called acetylcholine esterase. Okay, so and this is now finely tuned system. So basically, action potential comes in, acetylcholine is released, and then acetylcholine is chopped off. Okay, and certain number of these acetylcholines have to bind certain number of receptors that the muscle has its strength. Okay, if you have 100 acetylcholines, the strength of the muscle will be certain level. If you have 1,000 acetylcholines, the muscle strength will be higher. Okay, because more of this acetylcholine will bind more of the receptors. And as you can see, even in this case, you have one channel open, two channel open, three channels open, right, that allowing sodium in, but 1, 2, 3 are still closed. So if more acetylcholine comes in, more of them will be open, more sodium will come in, the higher shift of the membrane potential will be achieved towards the threshold so that the muscle can actually contract, okay? All this together is again called motor end plate or neuromuscular junction. Okay, so where is that neuromuscular junction formed? If you look at the top, right, we have this axon, we have this myelin with the Schwann cells, the axon terminals are coming in, and here is a new term or new anatomical kind of part or component that we have to learn is the sarcolemma, okay? So that's a new term that we have to learn about neuromuscular junction, and that sarcolemma is actually a cell membrane, okay, which surrounds or these muscles, you know, these striated, you know, fibers coming in, okay? And you can see here, when we zoom in on this, in this mid kind of panel, you know, the sarcolemma is here on the surface of this muscle, right? And the receptors are actually inside this sarcolemma. Sarcolemma is actually a membrane, okay, with these channels in, awaiting the acetylcholine to bind to allow the sodium to come in when it is bound. You can see the example, one acetylcholine receptor that has bound these two circles in this lowest panel, right, will allow sodium to come in, while the one receptor will be blocked or closed, and the second receptor will be blocked or closed because no acetylcholine is bound, okay? And just a reminder, that's a chemical, a neurotransmitter, you know, that is released by a nerve to bind to the postsynaptic terminals, okay? First disease affecting the neuromuscular junction, okay, that we will learn about is myasthenia gravis, okay? And here is what is the most important or giveaway symptom when a patient walks into your office, okay, and says, doctor, this and this, you know, when I do something, you know, my ability to perform that action, you know, gets worse as I do it or repeat it over and over again. And then if I take a rest, right, it improves, it gets better. And then again, if I reuse it, it gets worse and worse and worse. One example could be, let's say, blinking, right? first blink, it's very easy, you know, and then if I want to make second and third blink, it's much harder and harder to blink, okay? And then if I take a rest for an hour, I can again blink normally first time and then worse and worse and worse, okay? So that's very characteristic of myasthenia gravis, very specific for this disease. And now we're gonna kind of try to understand why is that the case, okay? So basically myasthenia gravis is considered to be autoimmune disorder. Okay, what does that mean? That our own protectors, right, that are protecting us against viruses, against bacteria, you know, against any intruders, right? Our own antibodies, right, that are floating around will start attacking something in the neuromuscular junction. Okay, so what is it? Okay, in this case, for some reason, the body in this patient will produce an antibody against acetylcholine receptor. Okay, in the left side, we can see the normal situation, right? We have this presynaptic terminal, acetylcholine in synaptic vesicles, the calcium comes in, it is released, and now all this acetylcholine can freely bind, you know, to the receptors that it can reach, right? And this will change the membrane potential, and contraction of the muscle cell will happen. But in the case of myasthenia gravis here on the right, what happens is antibodies will go and bind the receptors. And now if antibodies are blocking the receptors, where will acetylcholine bind? It cannot bind. It cannot allow sodium to go in, right? All the receptors, or many of them, are blocked, okay? So, and blocking of these receptors is actually preventing, you know, acetylcholine to induce, you know, the conformational change of the postsynaptic receptor to allow sodium to come in. And if sodium is not coming in, there is no change in the voltage, right? There is no depolarization. And for that reason, the muscle cell cannot contract, and that's known as paralysis, okay? Here is one example how paralysis happens, and you now know the concept or mechanism how it is happening, because there are antibodies blocking the receptors on the sarcolemma, on that membrane, right, of the muscle. All right, okay. So what are the symptoms? The first symptom is the eye muscle weakness. You can see here, you know, how this patient basically on the left eye cannot even keep eye open because it cannot stimulate, right, this muscle to open the eye, right? There is no depolarization of the muscle to contract to keep this eye open. But this is not the only case, right? Any muscle that we use for a movement, right, that can contribute, let's say, to eye movement, left, right, up and down, to smiling, to chewing, to talking, to swallowing. So wherever we have a muscle, you know, that is kind of being actively used, right, these receptors will be attacked wherever there is a neuromuscular junction, so to speak, you know. And then kind of the ultimate problem is if the muscles for breathing are affected, you know, these receptors for acetylcholine on the breathing muscles are being attacked, you know, it's harder and harder to breathe, and that can be then life-threatening or life-dangerous, okay? We are able to actually, patient is able to move first normally and then actually kind of wanes off, is when this acetylcholine actually comes out, right, and seeps into the synapse of the patient. We still have some receptors available. You see these ones on the left, right, available, and some in between, okay? Which means the first set of acetylcholine that will come in, right, will be able to bind these receptors that are available and induce the initial set of contractions, okay? However, you know, to remove them from the synapse, right, you remember we need the enzyme to work, we need glia cells to work, some reuptake to happen, and so on. So it takes time. So that means that all the other acetylcholines that are now coming maybe down with the second action potential, because we want to move for the second time, right, they don't have any more receptors where to bind, okay? And the second one will be worse and worse and will be harder and harder to initiate that movement, okay, because the available receptors are taken by first set of acetylcholine and by the autoantibodies, okay, so there is no more free receptors or sodium channels to open to allow influx in, to allow second kind of contraction of the muscle, okay. So hope that's clear. So that's why in this case, the movement actually worsens after the periods of activity. And then if the patient is taking the rest, it is actually allowing the time to remove the acetylcholine from the initial kind of rush or initial blast, right, by the enzyme or by the diffusion or by the glia cells. Okay. And now after some time, we have cleared out, so to speak, the ones that are available. And next action potential can come in and induce that contraction, okay? Okay, so in comparison to the myasthenia gravis, we have Lambert Eaton syndrome, okay? And that is a second condition that is known to be also associated by the autoimmune attacks, autoimmune problems that are attacking the neuromuscular junction, okay? So, but here is the difference, okay? The difference is that these autoantibodies in Lambert Eaton syndrome compared to the myasthenia gravis, okay, will attack those calcium channels, okay? Not the receptors, not acetylcholine receptors. The antibody will go and attack the calcium channels, okay? Which means that when the action potential comes down, okay, the calcium channels will not be able to open, right? However, all the acetylcholine that will come out will be able to bind to the postsynaptic terminal. So the idea here is, kind of initiate as many action potentials you can so that the calcium channels that are still available, allow more and more calcium to go in, okay? And to allow more and more acetylcholine to leak out, so to speak, released into synaptic cleft, so the muscle is induced into contraction. So why is that important for the symptoms? Because in this case, right, in Lambert-Eaton syndrome, we want patients to try to do more and more movements, because more and more trials means more and more action potential, which means more and more calcium channels open that are not blocked by the autoantibody against it in Lambert-Eaton syndrome. And in this case, the movement will improve with the repetitive effort of the patient, okay? Which is in contrast to myasthenia gravis, as you remember, right? We have, with the repetitive movement, worse and worse movement in the myasthenia gravis, while here we have more and more kind of movement with the trials one after the other by the patient, okay? And I hope that's conceptually clear, because in myasthenia gravis, what is taken up? The receptor, right? And there is a certain amount of real estate that we can use to contract the muscle. In Lambert-Eaton syndrome, the real estate is available. However, somebody has to release those acetylcholines, to get to them and induce the contraction, right? And that we can do by more and more action potentials, because you remember that more action potentials come down, more calcium channels will be open, and more neurotransmitters will be released, okay? All right. And the second difference is, as you kind of realize, that in myasthenia gravis, the muscle cells are being attacked by autoantibodies, and their connection is made with the thymoma, right? Okay. While in the Lambert-Eaton syndrome, who is being attacked? Neurons. The presynaptic terminal is being attacked, but the muscle is being preserved, okay? And the disease with which the Lambert-Eaton syndrome is being connected is surprisingly a small cell lung cancer, okay? And this is a high-yield question for not only for MCAT, but also for USMLE, okay? So now we have clear differences between the two diseases attacking neuromuscular junction. In both of them, the patient will have issues with the movement, right? In the first one, the myasthenia gravis, the disease is connected to thymoma, because antibodies are attacking the receptor, right, for acetylcholine. In the second case, in Lambert-Eaton syndrome, the disease is connected with the small cell lung cancer, right? And the antibodies are attacking the calcium channels, okay? The idea is, that the body is actually trying to fight the small cell lung cancer. However, by mistake, it also attacks the calcium channels. So now, since we have these issues, connecting the sound with the picture, you will have to kind of take this link and just copy paste it in the YouTube and watch it on your own consciousness and your own kind of will and time. All right. Do we have any questions? Okay, for today?